Well, it's a real pleasure to be here. I'm an oncologist from Auckland, and I'm also a researcher. And for me, research is part of my clinical care. You know, we don't accept that what we're doing now is good enough, and so we have to look to the future and try and make a change. Can I just acknowledge Rod on behalf of, if Unicorn New Zealand doesn't mind me grabbing this moment to say on behalf of the patients particularly that there's a lot of patients in New Zealand who echo the same sentiments we saw. One of them was on there, but there are many others as well, and you've affected not just Australia but also the region. So thanks very much. And there's only one clinician here who could describe neuroendocrine cells as women, and there's only one clinician who could describe treatment as dangly bits, but and it's not me. But thanks very much, Rod. I'm going to talk to you about. I've got just under 20 minutes, and I'm going to tell half of my talk's going to be not about genomics, but about how we got to where we are now, so that you can understand a little bit about the genomics as well. So bear with me. First, I'm going to talk to you about 5 revolutions of cancer care treatment. And the first one, and basically these are all steps that have enabled surgeons to us to do better than surgery. And Ben's going to talk about surgery shortly. But the first one, of course, is anaesthesia. If having an operation has been happening for hundreds of years, but being able to do it without pain, of course, is quite an advance. And here's Ben in 1870s on the left there with his sharp scalpel. And there's the anaesthetist over on the right with his anaesthetic, very high-tech at that point, with a little cloth with some chloroform on it. And John, I'm sure you do a little bit fancier technology than this today. But of course, it's a big step in cancer care because suddenly you can remove tumours without it hurting. Secondly, radiation came along in the 1890s. Breast cancer, 1896, was the first time you could treat a tumour with radiation. Hold it against the breast, the tumour shrinks, the radiotherapy goes in all directions, including through the hand of the person who's holding it there. And so there was a lot of but recognition that radiation can shrink cancer. And one of the first things was to try and direct the beams, and here's the first linear accelerator trying to do that in the 1950s, and I think this is the first boy to receive successful radiotherapy with a linear accelerator for a brain tumour. Now, of course, as you've just seen from Rod, we don't even have to worry about lining up the beams. We can use molecules on the cancer cell to take the radiotherapy inside the cancer cells and not anywhere else, so huge advances. The third advance is chemotherapy, and it'll make sense, I hope, in a minute as to why I'm telling you about all of these things. And chemotherapy was found, again, somewhat accidentally, and you know that nitrogen mustard gases were used with devastating effect in the First World War, and they were outlawed because of that in the Second World War by convention. However, the Allies didn't quite trust that scenario, and they did take a big ship full of nitrogen mustard gas across to Europe in 1941, and the ship parked in Bari Harbour, was bombed, and the gas spread across the Allied troops, and many of them died from their own gas. What happened in the survivors is that their lymph nodes around their body shrank, and their blood counts dropped. And so this observation was seized on by some scientists at Yale, and they thought, well, if it can shrink lymph nodes with a gas, maybe we could make it into a liquid, inject it into a patient with a lymph node cancer, lymphoma, and shrink the cancer. And sure enough, that's what they did in 1941. And you can see here, I don't know if you can see that very well, but you can see under here there's a lot of lymph nodes under this man's arms and in his neck. And 3 weeks later, they had all gone. Unfortunately, it didn't last, but nonetheless, here's the first proof that chemotherapy, then in the form of modified nitrogen mustard, could shrink cancer. And so from the 1950s onward, chemotherapy became a mainstay of anti-cancer therapy. But as with all of these treatments, who do you give it to? Is it going to work? How's it? Why would it work? And it's understanding in genomics, which I'll talk about shortly, that helps us with that. Molecular targeted therapy has really only been around for about 20 years, and it seizes on the idea that cancer cells grow because of growth pathways in cells, or because they have some special abilities that normal cells don't have. And these things, which are sort of medical terms or scientific terms for the different hallmarks that a cancer can have, so it can do these different things that normal cells can't do, that allow it to grow in a crazy way, form a lump, and spread around the body. And I'll come back to that in just a moment. But what you have to understand is that the way this works is that if you think about our bodies, we're an animal which has about 30 to 50 trillion cells in it, quite a lot. And a lot of those cells have nuclei. So of those 30 trillion cells, a lot of them have nuclei, and that's like a control center. And inside that control center is the recipe for all of the things that cell needs to do. Now, it could end up in any part of the body, so it needs to do lots of things. They're organized, those recipes are organized into chromosomes. There's 23 pairs of chromosomes, and you get half from your mum and half from your dad to make up a pair. And there's about 1,000 genes or 1,000 recipes on each chromosome, roughly speaking. So about 23,000 genes in every cell, so in a lot of different cells. And that recipe is written by DNA, and there's about 3.5 billion pieces of DNA code in each cell. So whenever a cell grows, it needs to split into two, and so that cell needs to copy all 3.5 billion pieces of code, which is organized into 20 to 23,000 recipes. And you can imagine that when that happens, mistakes get made. And when you've got something that big, even if you only make 0.001% of mistakes and 0.001% of the code, it's still a lot of mistakes. We can normally repair those, but it doesn't always happen. And if it happens that the mistake is made in one of the recipes that controls the way the cell grows, and that's how cancer happens. So chemotherapy works by disrupting cells when they're trying to divide. Molecular targeted therapy says, okay, where are the mistakes in that cancer? What genes are involved? Can we match that to a drug that will switch off that problem? The fifth revolution is immunotherapy. It's really exciting. It is a genuine revolution. And what it does is as Michael has already explained to us today, basically cancer cells get a cloak over them, which means the immune system can't see them and gobble them up. We probably, sitting in this room, are making cancer cells or precancerous cells all the time, but our immune system says, hey, that's not supposed to be there, and gobbles it up. What clever, successful cancers hide from the immune system, and these drugs actually don't treat the cancer. They just take the veil off when the immune system says, oh, there's a cancer there, I didn't even see it, and goes in and gobbles it up. And we know in neuroendocrine tumours that this works in one type at the moment, Merkel cell carcinoma, so a neuroendocrine tumour of the skin. It works particularly well. You can see here, this graph here, when the bar goes down, that's tumours shrinking. When it goes up, it's when they grow. And for the majority of people on this very small trial of 24 patients, the tumours shrank. And at the moment, it's early data, it was published in June this year, but it looks as though most of these responses are very long-lasting. And in fact, the only person we've treated in New Zealand, the cancer has completely disappeared and is staying away. So a huge change. So in summary, 5 revolutions. But the question is, which treatment do you use for which person? Which one's going to have the longest lasting effect? Which one should you avoid? Which people do you not need to treat at all? Just remove with an operation, or just watch them because you don't have to worry about it. If we could understand the cancer itself really, really well, if we could find out what are the gene changes inside that cancer that made it a cancer, to tell this, and we tell that story of the cancer, we would know either maybe this is one we can leave alone, this is one we need to treat with this drug or that drug, this one will respond to PRRT but this one won't, and so on and so on. Then we can really individualize the therapy. And so what I'm going to talk about is that we want to make these combinations work. We often use more than one. We've been trying for 170 years, and we've started to develop an arsenal of tools, but we don't really know when to use them. And how do you choose? So that's genomics. So we're going to choose with genomics. Michael described Australia as a small island in the middle of the world, but mate, you should come from New Zealand. It's like the analogy would be just a little rock off the side, you know. So, you know, you showed the figure of showing the anti-proliferative agents. We don't have sunitinib funding, everolimus funding, PRRT funding. We only have one chemotherapy funding, which is temozolomide and capecitabine. You know, which is really quite a stunning difference. We're really good with temozolomide and capecitabine because that's all we have, but it's a disaster for our patients, of course. So we've got a lot of work to do. So just with acknowledging that, but just also saying that New Zealand can do some things really well, and one of the things we can do is work together. So the idea is that if we take tumours, which look a bit horrible here at the top, we understand what's happening inside them really, really well by sequencing the genes inside them. We match it to a drug, one of the 600-700 drugs that are coming through on clinical trial at the moment that target exact gene changes inside the tumour, and then we can turn the cancer growth off. So we're called Network. It's a nationwide program. It builds on patients' experience, which we record in a registry, and our registry at the moment has just over 3,000 patients in it. We collect cancers, either fresh frozen at the time that they're being resected or biopsied, or from archived tissues. We're upskilling the clinicians in New Zealand. We have a video-linked national MDM, which people can dial in from anywhere in the country and get really good advice. And then we do deep genomics on the tumours to try and understand what's happening and what's driving them and how we can switch it off. So it's a national program. It's across New Zealand, and we use the spine of 6 cancer centres, each with its own university. And here's our group based in Auckland on the left, and our primary investigators around the country. Our really key partners, the Unicorn Foundation New Zealand, Malcolm and Siobhan, who are both here, who are amazing and have driven this project from the initial inception in terms of funding through to and an ongoing partnership. Great cultural advice, data managers across the country, ethicists in Dunedin, scientific advisory committee from around the world, our pathologists who do the heavy lifting in Auckland, surgeons collecting the tissue, sequencing facility, bioinformaticians, incidental findings committee who help us to advise when to give information back to patients. And you can see it's a really big network trying to do work on neuroendocrine tumours. And so I'm going to tell you 3 things. The first is that just about every net is different. Just as you're all different with your different experience, so is the genome of your cancer. So what we did was we thought, well, we know there's lots of different types of neuroendocrine tumours. You know, there's pheochromocytomas, and there's pancreas, small intestine, lung, all these different sites. We'd expect them to be different. So why don't we start with one type, really well defined, so just the pancreas, just resectable, just well differentiated, and we'll see what's in there, and we'll see what that looks like. So really clinically similar. And what we found, this is just looking at mutations, each of these circles represents one tumour that was resected. And you can see there are some similarities. So for example, these top guys here and a couple of others have no mutations that we could find. These 4 here have all got a MEN1 mutation. But apart from that, every tumour is different. And even those ones I showed you that are similar actually have different changes when we look at whether the chromosomes, that's those 23 big clumps of genes, are present or not. So the first thing is that basically everybody is completely different when we look at mutations. And the second thing is we have to think about not just one gene at a time. A mutation is a mistake in a gene, but actually what happens to the chromosomes? Are all of the genes there at all? And first, before I tell you about that, we can start dividing these up as to what happens. You know, are some tumours associated with good behaviour and some associated with bad behaviour? And sure, these are the ones that sort of behaved badly. They spread after they were resected. But they don't look that different, really, from the ones that don't. And these are the lower grade tumours, and these are the higher grade tumours. So I guess the bad players that we need to worry about are these ones that came back slowly, these ones that came back quickly, and these ones that were fast growing when they were removed and haven't come back yet, but we're worried about them because they were aggressive looking down the microscope. When we think about this, some of these tumours have also lost whole chromosomes. So remember I was saying there's 23,000 genes, and they're organised into 23 chromosomes, so about 1,000 genes per chromosome. Sometimes and there's 2 copies of each gene you get from your mum and your dad. Sometimes whole chromosomes have been lost in these tumours. And these people with these tumours, inside their tumour, there was the chromosome 11, that's the chromosome number 11 out of 23. Chromosome 11 had been lost in these tumours, and that's the same chromosome that MEN1 sits on. And so, but all of these guys did really well. No, their tumours haven't come back. None of them have come back. This group here are really quite different. They've lost 10 different chromosomes. So huge amount of the genes inside the tumour have completely disappeared. So it's pretty amazing that these cells can even survive as cells, but they're really quite different. And so I'll come back in a moment to what this all means. But the summary is that every tumour is different by mutation because it's got a different set of mutations and it's lost different chromosomes. So that's the first point. The second point is that it's even more complicated than that because neuroendocrine tumours, once they start spreading, change again. This is imagine that's the liver and this is the pancreas. This is a pancreas neuroendocrine tumour, and the mutation was in a gene called ATRX. Don't worry about what that means, but it was a gene mutation. And then it had lost the 3rd chromosome and the 11th chromosome. When it spread to the liver, it wasn't quite the same. It was only this part, so a subclone of the tumour that spread to the liver. And so even though it spread to 2 different locations, the aggressive part of this tumour seemed to be slightly different from the whole of the tumour. That might not matter if it doesn't change the way you treat it, but it's a signal that you have to think, oh gosh, what I removed from the primary tumour might be a bit different from the most dangerous part of the tumour, which is the part in the liver. Let's look at another example. Here is, again, a pancreatic neuroendocrine tumour, which had a mutation in MEN1, and it had lost 5 different chromosomes, so 5 of the 23 chromosomes. It spread to lymph nodes, and the parts of the tumour where that happened had picked up 2 new gene mutations. So the important thing here is that if you'd looked at the primary tumour and thought, how am I going to treat this, you may have missed a lot of the information because the aggressive part of the tumour, the part that had spread, had different gene mutations from the primary tumour. Or and so in this case, this is even a little bit more complicated because here's a neuroendocrine tumour resected from the pancreas, and it had no mutations. It had lost 2 chromosomes, 6 and 18, and it had also lost chromosome 11. At the same time, a lymph node was resected from near the pancreas, so a bit further away, and that had only lost 1 of those chromosomes. A liver metastasis was later resected, which had lost 2 of the chromosomes, but different to the tumour that had spread in this direction from this direction. And then it was realised, actually, there was a lump in the small intestine as well. And that was resected, and it had no mutations. And actually, when we look at the genes at the pattern of genes inside the tumour, it had actually started in the small intestine, spread to the pancreas, which had spread to the liver, and this one was a separate clone that had spread to a lymph node. So it's really complicated. And the genes, but the moral of this story is that the genes, the mutations, the losses of these chromosomes is different in the metastases, the dangerous part, from the primary tumours, and that makes it hard. Finally, I think what this means is that treatment needs to be individualised. We've shown that all of the cancers are different, and that those different cancers then change again when they start spreading. If we're trying to make some decisions about these folk, obviously the most important tumours that we need to worry about are these ones that have already spread or that we're worried about spreading. What information do we have here that can tell us what treatment to use? If we look at this tumour, this one has a BRCA2 mutation, and that signals us as oncologists that tumour may be more likely to respond to drugs that affect a particular type of DNA damage that's controlled by BRCA2. And so platinum drugs and PARP inhibitors might be useful for this patient. And we could try that in a clinical trial setting. These 3 patients all have a PTEN mutation. In fact, they have both the copies of PTEN are mutated. And that's a marker for response to everolimus. So these are people who might be the ones who would benefit from everolimus. So you could use this information to say, try that drug, and that might be more like a better first option because these tumours are particularly sensitive to it. So we've looked deep inside the tumours and we've found the problem that's causing the tumour. This one has the added advantage of having an MSH2 mutation, which is a mutation associated with response to immune checkpoint inhibitors, those new cool drugs. Only one here that we've seen out of all of these tumours that could respond, but nonetheless, by picking through the tumours, we've been able to say, this one used that drug, that one used that drug, and so on. And this red group as a whole actually have very low expression of a gene called MGMT, which is, which as an oncologist means that a drug called temozolomide, the temozolomide capecitabine, our only option in New Zealand, may well be that these tumours may be sensitive to that. So what we're able to do is to personalise therapy using this information by this deep biological understanding. All of this, of course, needs to be tested in a clinical trial because this is just biology, and what we are extrapolating from other tumour types, we need to try it in neuroendocrine tumours. So in summary, genomics, we've talked about the 5 revolutions and the fact that there's efficacy there, but we don't often know when to use the therapy, and particularly when not to use it. Every net is different, nets change as they spread, and we think that means that treatment needs to be individualised. Can I just finish with one comment, particularly pertaining to the role of the Unicorn Foundation and Unicorn Foundation New Zealand when it comes to research and genomic research as well. Really encourage you to share your experience with others, and that means getting on a registry, because we can learn so much from your experience, what happens to you, what treatments work for you, how you find all of that, and then we can compare that to the tumours themselves, looking at the genes inside it, the genomics, and seeing what treatments have been effective. Similarly, if you can donate your samples, if you feel comfortable with that, of blood tests and tumour samples, this is the future. So deep understanding of the cancers, matching it with your actual experience and knowing which gene changes match up to response to which medicines. And thirdly, of course, joining a clinical trial. These are all things that are going to help those that follow you, and that's the amazing thing about what I see in the Unicorn Foundation, people working hard to share their experience so that other people with neuroendocrine tumours can benefit. And finally, we have a really good relationship with the Unicorn Foundation New Zealand, who help us to ask the right questions. When we were talking about our original plans, they said, that sounds fine, but have you thought of this other idea? And we hadn't. And so we did, and we developed a circulating tumour DNA study. And then a philanthropist said, if the patients want it, I want it. And they paid for it. So it was not only did the idea come through, but also the funding. Talking with patients and patient advocacy groups helps us to discuss the right way to do things. What are the ethical implications of this test, of this research? What should we do? And we need your voice in that discussion. And it's also helped us directly to get funding to do research on nets. So thanks again to all of the people who are involved, particularly Malcolm and Siobhan, and all of the people in the Unicorn Foundation. I see a few other faces here from New Zealand, which is a wonderful treat. Have a great day today, and please feel free to ask me any questions if I can.